blood vessel and blood cells, but from a peculiar point of view, I will say very much from the biochemical point of view. In fact, the title of the symposium is From Basic Science to Application. It's a great pleasure and honor to welcome Vladimir Muzikantov as a keynote speaker for this plenary section of our meeting. And just few words about Vlad. We know each other since many years. Vlad is graduate in internal medicine, so he's a medical doctor, has a background with a PhD in biochemistry. Its main activity, its main contribution is on targeted drug delivery. And I will say that has done basic research, but very advanced application with a number of patents and patents application in this field. I just want to mention that Vlad is professor at the Department of Pharmacology at Penn. It's also as a secondary professor at the Department of Medicine, just to underline its close interest in the application we are just mentioning. Is the director of the Center for Translational Target Therapeutics and Nanomedicine and vice chair at the Department of Pharmacology at Penn. The title today is Vascular Drug Delivery Using Blood Cells. Please, Vlad.
Thank you very much, Maura. I thank organizers and the chairs. Will I sit? For this invitation. It's fantastic and fabulous to be in Urbina, second time in my life. And please, I'm a terrible public speaker in general, so give me mercy. I'm speaking in the second language, and it's second language for you too, so we both will have to bear. This was my introductory slide, and I will propel with this. And this is the lab, and people who really do the work. Juan Carlos Murciano, Sergey, Carlos, Ron, Colin, and other people were involved very much in development of this particular drug delivery approach. And I was blessed with collaboration with many scientists at Penn and around Penn. And I'm working on drug delivery, as Mauro said. And the major problem of many drugs is they don't circulate well, they don't go to the target place in the body. And in order to improve their pharmacokinetics, we now try to load them in the particles carriers, antibodies, which deliver these drugs to the site of preferred action. And the idea is very old. This is Paul Ehrlich, a Nobel Prize winner of 1903, sharing with Ilya Mechnikov for discovery of immunity. Paul Ehrlich discovered antibodies, and he proposed to couple drugs to antibodies to make magic bullets. And of course, that was not realized at the time of Paul Ehrlich, and it still is under development, the whole topic, the whole area. And drug delivery relies on many formulation of carriers. Liposomes were around for about 30 years. Polymersomes came aboard like 10 years ago. They're magnetic, dendrimers, quantum dots. There are many iterations. They all have some elements of design which are modular, such as targeting moiety or masking moiety. Polyethylene glycol chains are attached to drugs or to carriers or to cells to prolong their circulation and to mask them from taken up by reticular endothelial system and other defense system. Unfortunately, polyethylene glycol coating, which is called stealth technology, is not a panacea because, first of all, not every drug can be coated by PEG. If you over-pegylate your drug or protein, then you inactivate it. You can cover just 10% of liposomal surface by polyethylene glycol. If you do more, then liposome will disintegrate. And in addition to all these technical issues, there are immunological issues. In contrast to mouse and other rodents, humans have natural antibodies to PEG. So you cannot really make a drug delivery system based on PEG, which will circulate for a really long time. And the nature made this carrier system a long time ago. It's a red blood cell. It's a natural carrier. It's a carrier for oxygen delivery proteins, but it can be used for other things. And it's most abundant blood cell. It has prolonged circulation. It has very reasonable shelf life and can be used for transfusion. So about 40 years ago, Garrett Ehler and some other groups proposed to use red blood cells for delivery of drugs in body. The medical problem they start with was enzyme replacement therapy for lysosomal storage diseases. The recombinant and other proteins don't circulate well, and they don't get to the cells well where the action is needed. So Ehler proposed if you load these enzymes into the red blood cell ghost and inject, then they will go to macrophages, to lysosomes, exactly where they need it. And on this pioneering concept, many other enzyme delivery systems were proposed to either create a prolonged depot of enzymes in the bloodstream or deliver them to macrophages. And in addition, new ideas came aboard in early 80s for modifying red blood cells with fibrin or modifying red blood cells with antibodies to deliver to endothelial cells and other cells. And early studies in animals were rather disappointing because none of these drug delivery systems really did the job because they didn't circulate well. In addition to these technical issues, which are obvious, in the beginning of the 80s, as some of you might remember, the pandemic of HIV broke up, and all products which were related to blood were essentially unfavorable. That was at least two reasons why drug delivery field was not paying much attention to red blood cells as a carrier. Very few people, including Maura, worked on this. In addition to these problems, perceived or real, there were a problem of modification of red blood cells during drug loading. Because you touch a red blood cell, you attach something, you insert something in the red blood cell, and it might change its property, it might change biocompatibility. And if biocompatibility is compromised, then this modified red blood cell will get eliminated, essentially similarly to elimination of old or senescent red blood cells. They fix complement, they get taken up by phagocytes, and this process, if it's uncontrolled, might lead to very dangerous side effects such as cytokine release, inflammation, and hemolysis. So Maura and other researchers essentially excelled the technology, and I stole this slide from Iri Dell website of hypertonic loading of red blood cells with substances, with dexamethasone or with enzymes, and then resealing with hypertonic solution, after which you can inject these red blood cells into a patient, and they will circulate, and they will change pharmacokinetics and distribution of a drug in a very favorable way. And there are many clinical trials which go now on with Eridel and Eritech and some other companies for treatment of lysosomal storage disease, inflammation, immune disease, neurology, etc. So the list of potential sites of intervention or targets for red blood cell drug delivery and red blood cell targeting is essentially everything that is accessible to blood cells, which is blood itself, reticuloendothelial system, pathologically leaky vasculature, sites of vascular injury, and of course processes, pathophysiological processes which take place in the bloodstream. And theoretically, you can see how long and extensive list of potential applications of red blood cell drug delivery and enzyme delivery systems. So far, we were discussing essentially one technology, one strategy for drug loading into carrier red blood cells, which is putting them inside the red blood cell in the aqueous inner volume, which in this case, it's sort of most logical to use a red blood cell as nature designed it for hemoglobin. But it's not the only way for drug loading to red blood cells. In theory, you can bind drugs or therapeutics or prodrugs to the surface of red blood cell. And you can do it in two ways. You can either take red blood cells from the body and then chemically conjugate your drugs to the surface, or you can devise a drug derivative which has an affinity to red blood cell and which would bind to red blood cell surface. And then you can either load these red blood cells ex vivo or inject your drug in vivo in the bloodstream, and it will find red blood cells and circulate with them. And my lab works on the second strategy, which is coupling of drugs to the surface of red blood cells. And one of the drugs I was always interested in, the class of a drug, is fibrinolytics, plasminogen activators, which dissolve fibrin clots. This is a huge problem in developed countries because thrombosis leads to ischemic perfusion, and this is the most common cause for mortality and morbidity in developed countries. The blood coagulation, and it involves many mechanisms, platelets, coagulation system, thrombin, and they sort of duplicate and reinforce each other. And that's why there is no any single antithrombotic drug which would be 100% effective. You can completely block platelets and still coagulation will take over, and vice versa. And if you completely block all mechanisms, then the patient will bleed. And for prophylaxis, people use inhibitors of thrombin or platelets. And for therapy, when you need to dissolve clot, which is formed already, people use plasminogen activators, urokinase, UPA, or tissue type TPA, which are proteases. They cleave plasminogen, form it into plasmin, and then plasmin cleaves fibrin. These drugs have very limited efficacy and delayed onset because they don't circulate well. They have inhibitors, and only a small fraction of a drug, TPA or UPA, which is injected in the patient, works on the target clot. And in order to achieve the medical efficacy, you have to infuse such doses which are unsafe because good clots, which are mural hemostatic clots, get lyse too. And risk of bleeding and side effects include also direct toxicity to the CNS and some other delicate parts of the body. So because of those reasons, use of plasminogen activators is still fairly limited, and there are some applications which are unfeasible, such as short-term thromboprophylaxis after surgical procedures. And we thought that if we understand how these molecules work and use red blood cells for their carriage, maybe we can change and improve the situation. So plasminogen activators, they all are middle-size globular proteins, 50 kilodalton. They all are serine proteases or proproteases, which means that they need cleavage in order to get activated. And they all work as proteases by converting plasminogen into plasmin, as I said. They all have inhibitors, including PI1 and other serpins, which contribute to their inefficacy, because in addition to poor circulation, they get inactivated very quickly, which all leads to what I already discussed, high doses and unacceptable side effects. So the idea is that if you inject plasminogen activators, they will get cleared, they will inflict side effects in some side organs, such as CNS. They might induce bleeding because they will attack mural clots as well as occlusive clots. And actually, they will attack occlusive clots much less effectively because the perfusion and infiltration in occlusive clots is very ineffective. So those are limitations of free plasminogen activators. So now imagine if you can make a plasminogen activator which binds to red blood cells, then by the size of the carrier, the access to CNS and to mural clots will be prohibited. They will circulate for long enough time to get incorporated into nascent pre-occluding clots and then lyse them from within. So that was our concept, which we started with. And it was sort of a new life for an older concept of targeted fibrinolytics, which was based on single-chain fragments to fibrin and single-chain fragments to platelets, activated platelets. Other people, in particularly Edgar Haber in Harvard, and some people from South and some people from Germany, fused single-chain fragments to which bound to specifically to components of clot with urokinase and with some other antithrombotic agents. And the efficacy, unfortunately, in animal models was not great. And the problem was that these fusions don't circulate for a very long time, so they cannot be used prophylactically. But if you inject them after clotting, then most of the target is already inaccessible for these fusion proteins. So we thought that maybe if we use red blood cells as carriers, then we will resolve these problems. So first condition, of course, is to have a prolonged circulation of your drug and do not compromise circulation of carrier red blood cell. So in this case, we have chromium-labeled red blood cells, which are loaded with plasminogen activators. And you see that plasminogen activators loading do not compromise circulation of red blood cells. And in this case, we trace plasminogen activators, and you see that free plasminogen activators, which are triangles, are cleared very quickly, and circles, which are plasminogen activators bound to red blood cells, are cleared very slowly. And this was sort of a pharmacokinetics background for a technology which we developed, in which we bound plasminogen activators to red blood cells, injected in mice prior to clotting, and then by Doppler measurements and by other measurements, we saw that although the propensity for bleeding was much lesser than with free TPA, the efficacy of blood clot dissolution was much more effective than free TPA. And that was the beginning of our journey on this path, which we published in Nature Biotech and get a very nice cover of the journal. And since we continue that. And I will show you a few slides with the data, which I will sort of flash very quickly, maybe much more quicker than we all need to appreciate the specific numbers. But because many of those data are published, I sort of rely on the journals more than on my talk. So one potential area of application of these red blood cell bound plasminogen activators would be, of course, stroke. And in stroke, TPA itself is almost not used because it's not effective and because it's very dangerous for the CNS. The graph, these graphs show that injection of RBC TPA versus free TPA permits to dissolve microemboli in the brain of a rat or a mouse very effectively comparing with free TPA. And what it leads to a prevention of ischemia reperfusion injury in the model of acute thrombotic ischemia in mice. And in fact, the survival of mice, the upper right graph, was 100% after treatment with RBC TPA, and it was 0 after treatment with free TPA. So instead of improving survival, free TPA aggravated survival because it inflicted damage in glial cells. What's interesting that plasminogen activators, as many other drugs, have many signaling pathways in the CNS, including activation of LRPs and other receptors, which break down the blood-brain barrier and activate glial cells. And all this is counterproductive and, in fact, dangerous in the context of cerebral ischemia, hypertension, and inflammation. And pushing envelope even further, we've tested red blood cell TPA in a model of traumatic brain injury. This is a situation in which any propensity for intracranial hemorrhage would be lethal. And as you can see, standard TPA, free TPA in this setting always exaggravates or worsens the injury. What we hoped for is that red blood cell TPA will not exaggerate the injury. And in fact, we saw that it even improves the recovery after injury. And the mechanism of this we still don't completely understand, but apparently there are several mechanisms by which TPA acts in the context of cerebral ischemia, one of which is beneficial, it's thrombolysis, but many of them are essentially dangerous because they induce pro-inflammatory signaling. And RBC TPA is deprived of this pro-inflammatory signaling component because it doesn't have an access to the CNS tissue, but it has a more powerful thrombolytic beneficial component. So with all this was studied with a prototype, so to say. We took red blood cells, we isolated, we washed them, essentially as Mauro does. But then instead of his technology putting drugs inside red blood cells, we conjugated chemically drugs to the surface of red blood cells. So, but if you attach drug to the surface red blood cells anyway, then you can try to do this by affinity. So we created a series of fusion proteins which bind to relatively safe glycoprotein determinants on red blood cell surface, and they are fused genetically, recombinantly with proteins, therapeutic proteins, plasminogen activators, and some others. One particularly interesting plasminogen activator is urokinase, because in contrast to TPA, which is constantly active, urokinase is secreted as inactive proenzyme, which requires cleavage by plasmin in order to get activated. So theoretically, we can use urokinase prodrug as a long circulating sort of prophylaxis. Now, the problem is that the cleavage site on urokinase molecule, which converts it into active urokinase by plasmin, is located next to the cleavage site by thrombin. And thrombin cleavage inactivates urokinase. So if you want to deliver urokinase as a prodrug in the context of prevention of thrombosis, you shoot yourself in the foot because the main pathological agent, thrombin, will inactivate it. But fortunately, if you delete 2 amino acids which are between those 2 cleavage sites, then you convert plasmin activation site into thrombin activation site. And this recombinant fusion protein, a mutant thrombin now activatable pro-urokinase, circulates after injection. It binds to red blood cells. It doesn't hurt red blood cells. It circulates with red blood cells for a long time. It is always found on red blood cells. And then it dissolves clots after their formation very quickly and effectively, although urokinase itself does not. So in this particular fusion, we sort of converted our enemy, thrombin, into our friend, because now it activates prodrug to act exactly where the thrombosis happens. But thrombin is a very potent and, oops, what did I do? Okay. Thrombin is a very potent molecule. It has many activities. It gets activated by tissue factor in response to any tissue injury. And then, in addition to formation of clot by activation of cleavage of fibrinogen, conversion into fibrin, which is pro-inflammatory itself, it activates platelets, which, of course, aggravates thrombosis, and also ischemic reperfusion and inflammation. And then, in addition to this, thrombin activates platelet protease activatable receptors on endothelial cells, which convert endothelial cells in pro-inflammatory phenotype. So all these things are fairly harmful in the context of acute inflammation or thrombosis. And there is a molecule on endothelial cells, thrombomodulin, which essentially counters all these activities of thrombin. It binds thrombin, and it prevents thrombin from activating platelets or converting fibrinogen into fibrin. Instead, it makes thrombin to cleave protein C. And protein C, converted into activated protein C, is a very potent anticoagulant and anti-inflammatory agent which works on platelets and endothelial cells via the same receptors, PAR1. So we reasoned that in the situations when endogenous thrombomodulin APC axis is deficient, and this is unfortunately the setting very usual for acute inflammation, acute lung injury, ischemic perfusion, stroke. In those settings, we can supplement the APC thrombomodulin system with recombinant thrombomodulin fused with single chain, which binds to red blood cells. And these data, which are published now in Blood, show exactly that, that radiolabeled single chain thrombomodulin fusion binds to red blood cell, circulates for a much longer time than soluble thrombomodulin, generates lots more of activated protein C in response to thrombin challenge, and dissolves clots, or actually it doesn't dissolve clots. It prevents clots from happening, and that's why blood flow is maintained in arterial, in venous vessels much more effectively than with soluble thrombomodulin. In addition to those antithrombotic functions, thrombomodulin has potent anti-inflammatory functions. Part of them are mediated by activated protein C, by mechanism PAR1 dependent, which I already mentioned. And in addition, thrombomodulin itself, on the very end and terminus, has a lectin domain which binds many cytokines and inactivates cytokines. So we thought that maybe red blood cell bound or targeted single chain thrombomodulin will be also anti-inflammatory. And that's exactly what we see in these new experiments in mice, where we inject equimolar soluble thrombomodulin versus single chain thrombomodulin binding to red blood cells. And then we saw that even at very low dose, RBC-targeted thrombomodulin fusion is much more effective in prevention all indexes of acute inflammation in endotoxin-induced injury model in mice. And in the interest of time, I will skip these comments on this particular graph and show that's just a cartoonish version that if you inject either single-chain pro-urokinase or single-chain thrombomodulin, which bind to red blood cells, then you cardinally change their pharmacokinetics and their therapeutic features, and you might be able to attain short-term, immediate, effective and safe thromboprophylaxis and anti-inflammatory effects. And there, even for coupling of drugs to the surface of red blood cells, you can find or devise many strategies. Chemical conjugation on A, you can attach receptor or a binding protein or an adapter protein to red blood cell, which will bind your drug, and we've done this with urokinase receptor. And you can devise affinity targeting, such as single chain fragment or antibody conjugate. And for the last iteration, which is C, which is perhaps the most clinically translationable, a new list of questions arise, such as which epitopes on human red blood cells to use in terms of safety, propensity, particular additional advantages such as microenvironment for the drug on the surface. And now we use single chain fragments which were devised using phage display selection of immunoglobulins from a macaque library which interact with human red blood cells. And it's as translational as it can be because these are primate single chains which will induce minimal side effects. And our new data, which are unpublished, show that we do select some primate single chains which bind very effectively to human and some other red blood cells, and we fuse them with thrombomodulin, and it works. And now we, of course, cannot test them in humans, but we are testing them in other systems. So in the last 2 minutes, I want to make an injection sort of realism or skepticism, because I mean, I admire Mauro for his ability to translate things to the clinics, but there's a huge road, of course, ahead of us, because in essence we working against good odds. This is as biological as it can be, very difficult to standardize, very unfavorable by industry, and very unfavorable by regulatory agents. So I would say that in pathological conditions where other drugs can do, drug delivery by red blood cells will never be successful. But for acute conditions where no other drugs work and where the morbidity and mortality is about 50%, then this once upon a time injection of things which bind to red blood cell might work. And I finish this with a slide which alludes into sort of hybridization, future hybridization of techniques and strategies. There are nanoparticles and liposomes. There are red blood cells. They are different, but you can combine them. My friend Samir Mitragotri works with me now on binding nanoparticles to red blood cells, which completely changes their pharmacokinetics. You also make nanoparticles which are covered by red blood cell membrane fragments, which might be good, might be bad. In essence, there are two very different platforms for drug delivery. One is based on synthetic, another is based on natural carriers. And perhaps we are now entering the phase in which intercalation of those two strategies will produce very interesting and versatile arsenal of new drug delivery systems. And with this, 14 seconds and still counting, I thank you for attention.
Thank you, Vlad, for this very exciting presentation. I think that we have, I don't know how to stop this, but you were exactly on time, so perfect. I think we have time for a couple of questions. If there are comments or questions, please don't hesitate, and we have a microphone for you. Giovanni.
Thank you, Vlad, for very nice presentation. As I'm interested in human application on humans, so my question is, have you ever tried to get the opinion of Food and Drug Administration, for example, about what they think on the final product coupling red blood cells, so very natural cells, and ex vivo coupling with another protein that could be also another natural protein. So which is the regulatory thinking on the new potential drug that is outside this?
Not very friendly. varying from not very friendly to nasty. Well, and you can, objectively speaking, you take a biological drug, which is itself very difficult to regulate. Then you conjugate it with a thing which completely changes its behavior in vivo, which might affect the target cell. I sort of can understand why the attitude of regulatory affairs organs is not very friendly. But I hope that we will have products which not just change the pharmacokinetics of our drugs, but which change the mechanism they work. And some of the examples of our work illustrate this, because for some applications, TPA, you just cannot use the doses which allow the new mechanism to work, because this dose is lethal. In addition to those sort of relatively simple examples, there are many more sophisticated examples. When you attach things to red blood cells or load into red blood cells, you change the microenvironment of a drug. And this is, again, very especially important for biologicals. For example, red blood cell glycocalyx protects TPA from inactivation. Loading drugs, proteins into red blood cell changes immune response. There are many breakthrough futures which may be achieved. And I think that if we are steady and focused and with perseverance, sooner or later we will see red blood cell directed therapies working.
Yes.
Is there any difference in the lifetime of the red blood cell conjugated with respect to the normal red blood cell?
Yes, of course. A red blood cell just looks simple and sturdy and stupid because it doesn't have a nucleus, etc. It's a very delicate cell which experiences maybe the most extreme challenges in the body, because it squeezes through the capillaries and it flops over the heart valves, where shear stress is like in a hydroelectric station. And it survives because it's uniquely designed for this. When we touch a red blood cell, when we load the red blood cell or attach things to its surface, if we do it excessively, or if we do it in a way that compromises specific mechanisms which protect the red blood cell, we will screw the cell. And never a red blood cell loaded drug will circulate as well as native red blood cell. Now, in some formulation techniques, there is a sort of byproduct, maybe not very welcomed effect of hemolysis. Maybe it's not too bad, because when we deal with red blood cell from a donor, we deal with a mixed population of old red blood cells, young red blood cells, mature red blood cells. And if our system for drug loading selectively eliminates senescent red blood cells, then maybe our drug formulation will circulate almost as well as native.
Sorry, do you have any experience to use red blood cells to deliver, for instance, antibodies used in cancer therapies?
No, personally I don't. I know of a company in Canada which used to work with red blood cell fragments to modify with antibodies against cancer epitopes for drug delivery. But I don't think it went very well. And I'm a very simple person. I work with very simple targets which are right in the bloodstream. And with tumors, you need to get out of the vasculature, and you cannot rely on EPR effect in this process, and you need to stimulate extravasation of your drug delivery system, being it a red blood cell or a liposome or nanoparticle, in the site of pathological changes. And there is no sensing mechanism. It's not a phagocyte, which goes by gradient of cytokines or chemokines in the site of injury. So I would be rather skeptical of an idea of using antibody-coated red blood cells for drug delivery to tumors. Now, if you're talking about macrophages or other reticular endothelial system components which are accessible, that's a different thing. But that's what our friend Mauro is working on.
Okay, so thank you very much, Vlad, for this wonderful presentation. Thank you.
The next speaker is
We are moving from cells to the real vascular system. In fact, the next presentation is on vascular branching, especially in tumor angiogenesis. Please look.
Okay, thank you for the invitation. Today I will show you some of our recent results on podosome and the role of podosome in vessel branching, with particular attention to tumor angiogenesis. So, just a brief introduction on podosome. Podosome are adhesive and proteolytic membrane structures, and they were discovered by the group of Piercarlo Marchisio and Guido Tarone in the 80s at the University of Torino. And Piercarlo found for the first time these circular actin structures that are in the ventral side of transformed fibroblast. In the following years, in collaboration with Alberta Zamorin Zallone and Federico Caricari Scapio, he described also podosome in osteoclast and in the other myeloid-derived cells. So, but it's only in the 2003 that the interest for the podosome rise again, because Stefan Linder described the function of podosome in the macrophage migration and invasion. And at the same time, other scientists describe they describe the podosome in a cancer cell, in invasive cancer cell, and they called this podosome invadopodia or invadosome. So, but our organized podosome, they are not so different from the focal adhesion because we have integrin that bind the extracellular matrix and integrin-associated proteins and a core of actin and actin-binding protein. The main difference is that we found in podosome also metalloproteases, and so we have degradative activity. And the other hallmark of podosome is that they organize in cluster, small or large cluster. This is depending on the cell type, because on the macrophages we have a cluster of podosome all around the cells, but in osteoclast, podosome are organized in rosette, then become belt, osteoclast belt, and probably are a precursor of the sealing zone for the bone resorption. Also in endothelial cells, we have a rosette of podosome, while in cancer cell, the invadopodia are not well organized. So we know, and there are many publications that endothelial cells express rosette or individual podosome, both in the aortic, microvascular, and vein-derived endothelial cells. but the function of podosome in endothelial cells is completely unknown. So when some years ago, we observed that when we stimulate the endothelial cells with VEGF, the vascular endothelial growth factor, for a long time, 24 or 48 hours, the number of rosette increased, while the individual podosome, single podosome, remain exactly the same. So the idea is the podosome rosette related to the angiogenic process? But unfortunately, this is not too easy to study podosome rosette because it's a dynamic process, and the rosette quickly form and after disappeared. And we have also to use a different marker because there is no non-evocal marker for the investigation of podosome. So we have to constrain with cortectin, actin, or other marker, as in this example, vinculin, paxillin, and so on. Podosome rosette have a degradative activity, as you can see in this picture, and the rosette degrade the gelatin, and there is also an increase of metalloproteases active on membrane, as in the case of MT1 MMP. But one of the main concerns about the podosome is if it really exists in vivo. So we move to another model. This is an explant of murine aorta that we maintain ex vivo and then stimulate with VEGF. Then we open the aorta and lay it on the glasses for the observation with the confocal microscopy. We were able to find some rosette with the costaining with cortactin and effectin. And also in this case, VEGF stimulation increased the number of podosome rosette. So we move on another model, intact vessels, and we try to find podosome rosette in angiogenic vessels. And here we use costaining with laminin for the vascular basement membrane. And we identify as a rosette the costain with cortactin and effectin when they colocalize with the laminin, because the rosette are present only in the basal side of the endothelial cells. Then using these methods to quantify rosette, we take advantage of this angiogenic tumor model. This is a RIP-TAG insulinoma, pancreatic insulinoma model, in which we have a specific angiogenic stage. And as you can see, the number of podosome increase, reaching the peak at the angiogenic stage. So the first conclusion is that angiogenic endothelial cells and endothelial cells in vessel are characterized by podosome rosette. And the presence of rosette depends mainly from the stimulation with the vascular endothelial growth factor. So the question was about the mechanism of formation of rosette. We screened for the integrin, probably you are not able to see, but we found several integrin that colocalize with podosome rosette, but only alpha-6 beta-1 is the integrin that binds laminin. When blocked by a functional blocking antibody, completely abrogate the formation of podosome rosette. So we confirmed this data in other system. They found the blocking antibody anti-alpha-6 blocked the gelatin degradation, and also the silencing of alpha-6 reduced the number of rosette, Also in our model of explanted aorta, we found integrin alpha 6 in rosette. And here we take advantage of the knockout model of integrin alpha 6. Then we expand the aorta from these mice. And as you can see, the number of rosette in knockout mice is reduced compared to wild-type mice. We treated also mice, the RIP-TAG mice, with a function-blocking antibody. We injected by tail vein. This antibody localized on vessel and stain the rosette. The treatment reduced the amount of podosome rosette. We confirmed this result also in a different model. This is a B16 melanoma tumor transplanted in syngenic mice, knockout for integrin alpha 6. Also in this case, the number of rosette is reduced. So we wonder what is the role of the ligand of integrin alpha 6 laminin in this podosome rosette formation. Unexpectedly, we found that laminin inhibit the formation of rosette. So increasing concentration of laminin completely abrogate the formation of rosette. So when we try to confirm this data in vivo, we found that there is a laminin knockout model. This is a laminin alpha 4 chain knockout mice. Laminin alpha 4 is the component of alpha 4, 1, and 1. This is the formerly called laminin 8. It is one of the laminin of the vascular basement membrane. Also in this model, we found that with the lack of laminin, we found an increase of podosome rosette. So this is difficult to explain, but when we performed this experiment with alpha 6 GFP by turf microscopy, and we found that alpha 6 decrease quickly and then appear again on the membrane, while in presence of laminin, the level of integrin alpha 6 remain exactly the same. So our hypothesis is that probably the integrin that form a podosome derive from the integrin in the focal adhesion, so bound to the laminin. This experiment with vinculin RFP in turf microscopy confirmed that this hypothesis. We have not yet formal and direct evidence that the integrin or other component of the podosome come from the focal adhesion. But some experiment with nocodazole and primokine drugs that block the endocytosis of the focal adhesion and the recycling of the component of the focal adhesion demonstrate that probably integrin required to be trafficked to the early endosome and then goes on the membrane on the podosome rosette. So the second conclusion is that integrin alpha 6 is required for the podosome rosette formation, and the laminin inhibit the formation of rosette by blocking the translocation from the focal adhesion of the integrin alpha 6. But what about the vessel branching? Probably you know that tumor vasculature is very different from the normal vasculature. One of the hallmarks of the tumor vessels that are hyperbranched, so they form too much new sprout and new vessels. So the model, the mechanism that regulate, could control the formation of sprout and the vessels is well studied, and is mainly dependent from two different molecular mechanisms, VEGF and VEGF receptor 2, and the other system is the DLL4 and Notch system. Because it has been demonstrated that quiescent endothelial cells respond to high concentration VEGF, and some cells become tip cells, change phenotype with philopodia, and migrate in the extracellular space. This cell produce high level of delta ligand 4 that activate Notch in the neighboring cells, and these cells are inhibited to form new sprout. With this mechanism, vessel form regular new sprout, and so new vessels. And in tumor, this mechanism is altered. So what is now not known is the role of the basement membrane in this model, because the basement membrane is difficult to overcome for endothelial cells. Easy for cell derived from the hematopoietic system, but not so simple to cross for the endothelial cells that have to degrade the vascular basement membrane. So our hypothesis is that podosomes are involved in the process of basement membrane degradation and in the sprouting of new vessels. So some years ago, we set up these angiogenic assays that is small pieces of murine aorta that are explanted and embedded in collagen gel, and they form new capillary-like structures. So we look for podosome rosette in these capillaries, and we found this rosette, but again, unexpectedly, these rosettes are not in the tip cell, but are far from the tip cell, so in the distal part of the capillaries. So the idea is that probably they are involved in the formation of new sprout far from the tip cell, where the basement membrane is already present. So to demonstrate this, we set up this experiment with multiphoton microscopy. And as you can see, when rosette appears, a protrusion were formed. And some of these protrusion could form new vessels. These experiments are also supported by not a direct evidence in alpha-6 mice that show a reduced number of branching, also in the laminin knockout mouse that express a high number of new vessels. And no, as you can see here, in a normal capillary branch, a regular time, and while while in the knockout mice, they form more linear structures. So, and finally, we perform other experiments with the blocking antibody in RipTag mice. And also in this system, we found a reduced number of new sprout, new branches, as also in the melanoma that I showed you before. So, in conclusion, the model that we propose that when the endothelial cells are in a quiescent vessel, the high level of laminin maintain the integrin alpha 6 in focal adhesion and not in podosome. When the high level of VEGF induce the expression of integrin alpha 6, integrin alpha 6 move to podosome rosette, then degrade the vascular basement membrane, and this then allowing the sprouting on the tip cell and the formation of a new vessels. So, and this is the group that work on this project, and a special thanks to Giorgio Seano, who performed much of this experiment. So, thank you for the attention.
Thank you, Luca, very much for this wonderful presentation that is now open for discussion. Any question or comment to Luca? Yes, Vlad.
A very naive question. Do any of this process also involve in transmigration of leukocytes? I mean, it looks as by Dejana works that some fraction of leukocytes go not between the endothelial cells, but across the endothelial cell. And apparently this should involve some interaction, dynamic interaction of basal laminal surface of endothelial cell with a basal membrane, and guidance from the side of leukocyte by MMPs or some other. I mean, do you see any commonality in what you study with what I'm asking?
This is an interesting hypothesis, because we wonder if leukocytes use podosome rosette to cross the endothelial cell in transendothelial migration. We have no data for the moment on this. We never see any leukocyte in the vivo experiment, but I think we have to look with attention for this. Yeah. Yeah.
Other questions or comments for Luca? Can I ask what kind of intervention, or in other way, what can you block to differentiate the sprouting from tumor system with respect to the normal endothelial growing?
So
you mentioned VEGF, but
yeah.
If you block VEGF availability, can you differentiate the intervention from?
No, the idea in this case is that we have not to block VEGF, because VEGF is important for many other aspects. Yes. So blocking podosome or specific mechanism in the sprouting, we can obtain a sort of normalization of the tumor angiogenesis. And today, one of the aim of these studies in the field of tumor angiogenesis is normalize the tumor vasculature. Several people, Rakesh Jain, for example, think that it's better to normalize the vasculature than to eliminate the tumor vasculature, because it's easier targeting the tumor cell with drug or with the radiotherapy, and because the level of oxygen is better than in hypoxis environment. So.
Yes. Any comment or question for Luca? If not, thank you very much again for your presentation.
So we moved
I would like, first of all, to thank the organizer for giving me the opportunity to show you some results about the leg ulcer microenvironment. So it is well known that chronic venous disease is a very widespread pathology among women especially. And the stages is very important to highlight how the progression may be identified through biochemical markers. So the different stages that we know and we review in the recent guideline start from C0 with no clinical signs, through varicose vein, the well-known among women, and the active venous leg ulcers. And it is maybe important to obtain information about the possibility to discern what are the main biomarkers that may discern the different stages among the progression of the pathology. And it is well known that from a histological point of view, the varicose vein is characterized by the altered endothelial cells and the extracellular matrix condition that may be obviously altered also by cell biology point of view, in which we recognize the identification also of the recruitment and the rolling of several leukocytes to the endothelial cells. It is important to note that also from a biochemical point of view, in this stage, we recognize the secretion of several proteases, matrix metalloproteinases, that able to almost degrade the extracellular matrix component, but also the presence of several cytokines, inflammatory cytokines, that may be able to induce the degradation of glycosaminoglycans, that is a layer that protects endothelial cells. So the degradation of the glycosaminoglycans layer prone the endothelial cells to the activity, to the deleterious effect of matrix metalloproteinase and cytokine. The last step is the chronic venous ulcers, the active condition that may be recognized from the histological point of view with complete loss of epidermal and dermal matrix component, but also from a cell biology point of view, consistent, significant tissue infiltration by mast cells and macrophages that may be able to also induce, from a biochemical point of view, of several proteinases among the matrix metalloproteinase, activators of plasminogen, and so on, but a really wide expression of cytokines, inducing the consistent and significant abolition of the protective effect of the endothelial cells. So it is well known that the chronic venous disease or chronic venous insufficiency may be based on two main aspects: the shear stress and the hemodynamic complex complications in this case, but also from several conditions with proning conditions, sanitary, metabolic disorders, genetic mutation, and so on, prone the endothelial to active dysfunction, loss of glycocalyx protecting effect, inducing the endothelial activation, so proning also the cells with the edema, blood stasis, ulcers, chronic reflux, typical of chronic venous disease. But we have pointed out our attention to mechanisms that may be identified through the modification and the transformation of circulating monocyte in macrophage, inducing inflammation, inducing endothelial dysfunction, releasing cytokine and chemokines, several protease growth factors that are able to induce ulcer microenvironment. So we would like to point our attention to how inflammation and several biomarkers may be useful to discern what kind of biomarker may be useful to identify healing condition or non-healing condition in ulcers. So studying the venous ulcers microenvironment may be really important because we have the we have some biochemical basis in which we identified in chronic condition that inflammation play a crucial role, and may be important to also identify what kind of biomarker may act as a switch on the inflammation or switch off the anti-inflammatory pathways. We have also identified in ulcer fluid composition several components, and we pointed out that in cytokine and matrix metalloproteins and their tissue inhibitors are specific to identify a balance between the degradation or the anti-proteolytic effect of this compound. We have analyzed a plethora of ulcer wound fluid, and we separate the samples obtained from these women in inflammatory and granulating. Inflammatory is non-healing condition, and the ulcer is not easy healing during time. Respect to granulating ulcers with healing condition that within 3 or 4 months close the wound by this condition. So we have identified among 65 biomarkers that in non-healing condition we have identified the interleukin-1 beta, interleukin-12, interleukin-8, and vascular endothelial growth factor much more expressed respect to the healing condition. So this may be useful to identify non-healing condition a pro-inflammatory microenvironment. On the other hand, we have also identified that in healing condition, we found IP10, RANTES, and platelet-derived growth factors higher express respect to the non-healing condition. So may be useful to identify also these parameters to discern ulcers that may be more easily prone to heal the ulcers in much faster time during the progression of the disease. On the other hand, we have also pointed out our attention to matrix metalloproteinases, and we have found that the gelatinase expression MMP2 and MMP9 and the elastase MMP12 are highly expressing during inflammatory condition, non-healing ulcers. On the other hand, we have also found that MMP1, MMP7, and MMP13, stromelisine, collagenase, and other component of the family of matrix metalloproteinases are highly expressed in healing conditions. So we have identified a balance between several matrix metalloproteinases that may be differently expressed during ulcers in healing and non-healing condition. For the first time, we have also identified them. TIM4 is highly expressed in healing conditions. So the higher expression of the tissue inhibitors of metalloproteinase 4 may be useful to downregulate the inflammatory condition and to stimulate the healing of some ulcers. On the contrary, TIM1 and TIM2 are highly expressed in non-healing condition, and so it is difficult to balance the higher proteolytic activity presence in some ulcers microenvironment. So, taken together, our results may be useful to identify a biomarker profile that may be useful, at least in some patients, to identify the 2 main phases of the progression of leg ulcers. The granulating phase are characterized by IP10, IP1, IP7, and so on. And the inflammatory condition of the ulcers are characterized by the classic inflammatory cytokines, and degradative gelatinase activity, and the classic molecules that be able to recruit macrophage and so to induce much more inflammation in the microenvironment. But we have also identified a plethora of cytokines and a component that are common in the ulcers, so are not able to discern through this, only these two, what kind of molecules are expressed by endothelial cells, macrophage, fibroblasts, and so on. So this is the mechanism basically expressed during the microenvironment of cells. We have also pointed out our attention to a particular condition. We have highlighted that the disappearance, the loss of glycosaminoglycans prone the endothelial cells to be much more easily subject to inflammatory conditions. So we have pointed our attention to glycosaminoglycans, sulfated and non-sulfated, and we have also used some of our experiments and our time to identify and to isolate and purify a component named sulodexide that is a highly purified glycosaminoglycans component by 80% of fast-moving heparin and 20% of dermatan sulfate, under patent in the USA recently. And this component is very useful because it has got antithrombotic, anti-inflammatory, and profibrinolytic and anti-lipemic properties. But we pointed our attention to the use of this compound because we would like to show how sulodexide may restore the glycosaminoglycan lacers and may be useful to stop or downregulate the inflammatory properties or increase the anti-inflammatory properties. So we have performed also some experiment in vitro, and we found that sulodexide is able to modulate biomarkers expression in THP1 macrophage cells under the stimuli with inflammatory lipopolysaccharide stimuli. And we have found that during inflammation processes, there is a plethora of component in red that may be found significantly increasing THP1. But if we treat, or better, pre-treat THP1 macrophage with this component of glycosaminoglycan, we found a significantly downregulation of several compounds to the normal and increase of anti-inflammatory properties of both MMP and cytokines. So this may be useful because also sulodexide may be able in vitro in adult endothelial cells to when pre-treated at the concentration clinically used with these commercially available drugs, is able to almost complete the scratch that we have performed in laboratory. So the use of glycosaminoglycan sulodexide may be able to induce proliferation and accelerate the wound healing in vitro. Another possibility to obtain information is what is our take-home message. We have identified the inflammatory phase of leg ulcers with respect to the granulating phase of leg ulcers. And we have also identified that the differentiation of monocyte into macrophage may be obtained through the identification of a plethora of components in cytokine, chemokines, and also matrix metalloproteinases. It is interesting to note that it's noteworthy that sulodexide may be able to induce not only the component, but selected component. So we have also identified that among these components, sulodexide is able to induce the downregulation of inflammatory cytokines and matrix metalloproteinases, and to downregulate also the component useful for granulating phase of the healing condition of the ulcers. So we take this opportunity to hope that sulodexide may be a drug that may act as Janus bifrons effect, inducing the downregulation of inflammatory condition and upregulation of granulating conditions. So we hope to have identified the mechanism by which sulodexide may be able to switch on the granulating phase in wound healing and to switch off the inflammatory phases of this pathology. We would like also to thank the organizer for giving me this opportunity, and all the collaborators in Urbino, in the USA for the preclinical studies, and for Luca Giovanni Mosti for the collection of the samples. Thank you to you all of you.
Thank you, Nando. I think we have time for a couple of questions or comments. Yes, Alessandra.
It's very interesting. I have a naive question about the PDGF, of course.
Yeah.
But so if I understood well, the patients that are more prone to healing have an increased PDGF in the supernatant of the ulcers.
Yeah.
So what does it mean that, why? So because there are more platelets, or platelets are different from one patient to the other?
I don't know. Really, I don't know. We have found that platelet-derived growth factor BB is highly expressed in healing condition. So we would like also to perform in venous from varicose vein, in edema, in different steps of the progression of the disease. And I would like to know if platelet-derived is only secreted in the ulcers condition or may be present during the progression. So because platelet may be also intricate with endothelial cells, the possible role of the joint link, I don't know. Actually, I don't know. I'm sorry.
Just a second brief question. And what is known about exogenous PDGF? Is it the platelet gel is used for this kind of ulcers? Or?
There are several trials that may try to use the platelet-rich plasma, but is not so easy to use or so useful for the healing condition. Is a matter of question. Is a matter of question.
Thank you.
Okay, other comments or questions from the audience? If not, I thank Nando for this presentation and all the speakers from this presentation. Thank you very much for contributing to this section. Now it's time for coffee. We have 30 minutes.